Yeah, so most of the work I do is modeling various aspects of coral reef ecosystems. So I'm sure most of you know, but corals are, oh, they're cnidarians, but they look a lot like rocks. Coral drawing. But what's really kind of interesting and complicated about them is kind of like the lichens we learned about yesterday. They're kind of two parts. So they're the coral animal, but inside the coral are these little symbiodinium algae. And these algae photosynthesize. They provide the coral with fixed carbon. And the coral, in response, provides the algae with a home and also with nutrients. And corals, they're important because they build coral reef ecosystems that are super diverse and productive. But they're also really, really threatened by climate change, in particular, increasing temperatures, ocean acidification, eutrophication, all that. And so a lot of people are working on studying how corals are responding to these changing abiotic factors. And so to do that, they put the corals, in tanks in the lab, and they measure different responses. You can increase the water temperature, you can increase light levels, things like that. And this is really cool. I don't do any of the experiments, that intimidates me, but I do the modeling, and these types of experiments, these physiological experiments, lend themselves really well to bioenergetic models. So I've worked with a type of bioenergetic model called dynamic energy budget models, or DEB models. So DEB models are really cool because they really explicitly track all the flows of materials and energy within an organism, or in this case, two organisms, because you have the coral and its algae. So you could have the coral here. I'm just going to call it the host because C for coral and carbon is confusing. And then you have the symbiont. So the model can track, for instance, like the input of light and how the symbiont then photosynthesizes, and the amount of fixed carbon it shares with its host. And then the host, in return, you can model the amount of nitrogen that the host shares with the symbiont. You can also model, inputs of environmental nitrogen to see how that, moves through everything. And then the output of these models is typically something like S growth, so like change in symbiont. Sorry, my writing is terrible, but and host growth. So change in the biomass of these different organisms over time, which is super cool because then you can use these models to look at things like coral bleaching, which is the phenomenon when temperatures get really high, the symbiont, they start producing a lot of reactive oxygen species, so the coral kicks them out, it loses its color, turns white, and if that persists long enough, the coral can die. As you probably heard, there's been a lot of mass bleaching events with these heat waves we've been having, so models like this are very useful for that. But coral reefs are super diverse, so there's also all these community level interactions that the corals are participating in. So in particular, one that a lot of people care about is the interaction between your coral and then the macroalgae. So there's a lot of different species of macroalgae that live on reefs, and they compete with the corals for space on the reef. And if the corals are stressed, or if a bleaching event occurred and a lot of them are dead, the corals can outcompete the algae for space, and you can get this transition to algae dominance. And so this is also an important phenomenon. There's been a lot of models that have looked at this. They're usually relatively simple models, like ordinary differential equations, like the kind Holly talked about yesterday. So you'd have the coral growth, you'd have the algae growth, macroalgae, not to be confused with the algae in the coral. And then you'd have, like some competitive effect of the algae on the corals. And then you could have, your T and have all of that, and the MDT, and have just your basic, growth, mortality. And these models, they're relatively simple, but they predict really interesting dynamics, in particular by stability between the coral and macroalgae states. So they predict that a coral-dominated reef or a macroalgae-dominated reef under the same environmental conditions could both be stable depending on the history of the system, which is really important because you'd care if, like, all of a sudden your reef flipped from coral-dominated to macroalgae-dominated, because generally we prefer the coral-dominated reefs, and so do the fish. So the question I have is that if you have these dynamic energy budget models that are really closely tied to experimental work, is there a way that you could like kind of integrate them with the community level models to then tie in these community level dynamics to the abiotic, like the physiology that's happening inside the coral, and then use that to predict, okay, if the temperature is changing, light levels are changing, nutrients are changing, how is that then gonna, propagate up and affect the ability of the coral to compete with the macroalgae? And how would you do that without ending up with this super giant, really intractable model? And how would you deal with like the different time scales? Because obviously this is happening on super small time scales. This is happening, months or years. Yeah, so question that's not answered. Yeah.
Second time scale that you're talking about of like this long months, years, decades, maybe, that is in part set by GC being slow.
Yes.
Like the coral requires mineralization.
Yeah, exactly. So corals, they have the tissue growth, but then they also have the calcification, and that's much slower. It depends on the species. Some species can actually be pretty rapid and grow, in a couple months, but others, it takes them, they're hundreds of years old and they're these giant mountain corals.
Oh, interesting. Good.
So when you have the competing of the algae with the coral, is the algae competing with the coral, or is it competing with the algae in the coral?
Oh, good question. So part of it is a shading. So if this algae grows like over this coral, then I guess it's technically competing with the algae in the coral for light, but the result is that this coral doesn't grow. And then there's also like physical or chemical competition between the algae and the coral animal itself. So some of the algae produces like noxious compounds that can kill off the coral tissue, or, like physical abrasion of the algae on the coral itself.
I know you just said it's an unsolved problem, but this question of bridging models that act at different timescales without making you're just so complicated that it's not fun anymore. Like I run into it too in a very different way. I'm just wondering like how you think about like the first step towards figuring out how to figure it out.
Yeah. So when I thought about it, I kind of thought about focusing on the energy part and seeing how much you could simplify that. And the time scale I haven't thought as much about, but the two model part, like if there's this, it's kind of hard with the coral because there's the two parts, but if there's any way to see if you could like strip down the bioenergetic model to just like the really essential components that have the biggest impact on the resultant growth, and then put that into like the GC parameter.
Nice talk. I really like this kind of two temporal scale. So I was wondering if you or others are interested in like the spatial dynamics here, and if that's something that you can get data on for these systems.
Yeah, definitely. So I've done some spatial modeling of this part of it, like partial differential equation. And like I've worked with data from Moorea, French Polynesia, because there's the LTER at Santa Barbara, where they have like a robot, and they like put the robot on like, I don't know, like a boogie board, and the robot goes over and takes pictures at like really frequent intervals. So you get this like really high spatial detail of the coral and macroalgae components. For the smaller spatial scale, like within the coral, I think there is a lot there because, like the top of the coral has more light. So there's more photosynthesis happening there than at like the bottom. But I haven't done any work on like the smaller spatial within a coral.
Can this model factor in the competition between algae and corals and corals?
Oh, that's a very,
area.
Yeah, that's a very good point. I think this basic level doesn't. I have seen some that like include sponges, but there's definitely, there's different types of coral. There's the slow growing like almond corals. There's the more weedy like branching corals. And I think at this point, people don't care. Like if you have corals competing with coral, that's a good thing because you have a lot of coral. So it hasn't been as much of a focus, I think, but you definitely could. And it would be important if you cared about like the composition of your coral community, because they have different, like the branching corals are habitat for fish, the mounting corals give more like coastal protection, and they kind of really build the reef foundation. So yeah, good question.
Sorry, second question. On the DEB model, do they assume it's just one algae or multiple species of algae that are, so then I'm assuming there's tons of different species of
Yeah.
algae that are interacting.
Yeah, really good question. So in the simplest version, you just have one species, but so like one algae at a time in the coral, but you can vary like what the parameters are for different species. So yeah, there's like a whole bunch of different Symbiodiniaceae algae, and they have different properties. Some are like more fast growing, some are like better in heat. I have collaborators who did do a model that they expanded on this, and they had two different types of Symbiodinium, and there was some interesting outcomes with how those two competed within the coral and how the coral like differentially allocated nutrients to each of them. So there's, yeah, definitely lots of interesting questions there. And then like the whole symbiont shuffling thing where corals can like kick out some and take in others.
Well, thank Raine again. That was awesome.